Okay, so we left off in the last video introducing this idea of the ways that we're going to protect you. And I say the three phases are based on the timing of effects, right? So your barrier defenses, right, are instant and constant. And this includes your skin and mucous membranes, right? The things that line, right, all the entries to the body and throughout the body. So mucous membranes are what contact the outside world, and skin is what contacts the outside world. So this contact is always bombarded by potential pathogens. Innate immune defense, all right, so innate means I'm born with this. This is part of me, right? So that's why this is rapid. But I say very importantly, it's not specific. So it doesn't really know what the specific pathogen is. It just knows there is a problem. And this is going to be part of the cells, right, and soluble proteins, whereas barriers really are the first line of defense. And so soluble factors means, again, mostly proteins, right? So stuff that stays in solution in the bodily fluids, right? And then third is the adaptive immune defense, the third line. And this is cells and soluble factors, but here we're talking about antibodies. We're going to limit those soluble factors. This, of course, is the third because it doesn't happen as quickly. It takes more time. And this is specific. And note here, you're not born with this just ready to go, so that's why it's slower as opposed to this one, right? But adaptive means that adaptive comes from the specificity, that these cells, right, here's going to be B cells and T cells and antibodies associated with them, are going to be able to really identify that first thing I said in the last video. You've got to be able to, right, recognize the problem if you're going to get a defense. And so these guys are able to recognize very specifically, and they can change their defense based on that. These guys will never change their defense. The defense will be mildly adjusted for the amount of damage and inflammation or the degree of problem in fever, but really it's not going to change to suit what's going on, as the term adaptive means. All right, so here's three classes of cells, because we've said barrier cells and soluble proteins. So our cells come as phagocytes, lymphocytes, and cells with cytoplasmic granules, which is very good. We've learned about some of these in the past. In fact, we learned about them in blood. So this is why you're looking at the family tree, if you will, of blood cells, right? That's starting with that hematocytoblast and producing all these cells. So we're going to ignore these two in this right now. We're looking at these guys in this area. And so the phagocytes are those that basically will eat, right? Because that's what phago means, the cells that eat. So they're going to just consume pathogens to destroy them, which is pretty effective. Again, but they don't really, the phagocytes we're going to talk about here do not know. They're part of the non-specific. So we said that all the phils were actually phagocytes, though neutrophils are the most effective. And then, of course, monocytes, right, are really good because they become macrophages. Lymphocytes, which are over here on their own branch, appropriately so, because these are part of the specific, with the exception of this one. That one is not part of the specific. They are going to be able to, right, be the specific adaptable. They're going to have what we call antigen receptors, and that's the big deal. And it is a big deal. And they're going to be able to produce memory. Huge deal. All right. And then there's cells with cytoplasmic granules, all right, which is really what you were studying in the blood cells too. And there's a lot of these. The natural killer cells have these. All the phils have these, right? And so these are the visible ones, and even T lymphocytes have some granules in them too. But here again, cells with cytoplasmic granules. Those granules are chemicals, and this is chemical warfare. And these chemicals are going to do things from range from killing something to calling other cells into the area to increasing blood flow to the area, right? So you are used to this from the whole process of clotting that we talked about in blood. All right, so here the lymphocytes, again, these are part of the specific response. They are the primary drivers of the specific response. And we also, again, call the specific response adaptive because it changes according to how you change. So these are known as B and T cells. And you couldn't tell the difference when we looked at the slides. There wasn't any way to distinguish this visually on the magnification you have. But they do differ, if you had really high magnification, in what kinds of proteins they have on their surface. And they differ in the molecules that they use to mediate this response. All right. Furthermore, they have differences in where they mature. So they are both born, like we saw in this situation, in the red bone marrow from that hematocytoplast. But they take some different paths as they become developed, and this process of developing, changing gene expression, we call maturation. All right. But both of them will develop, both of these B and T cells will become specific because they will develop receptors for antigens. All right, so here I say separate the B cells, primarily defend you by producing antibodies. Antibodies are finders, and that's how you should always think of them. Antibodies are proteins that find antigens. Now, antigens are chemical structures. Again, a lot of antigens are proteins, like the antigens we talked about on the for the ABO, for the blood types, and the RH factor, but not all. So again, that's why we're using this general term, chemical structures. So antigens are chemical structures on the surface of pathogens, so in the plasma membranes of these. And these, again, antigens are identifiers. So we have the finders and the identifiers. So B cells will make these finders, and they'll make a lot of them. T cells secrete factors, again, chemicals, to enhance or destroy cells that are infected with pathogens. So T cells are also going to have receptors for antigens. They will, in some sense, find, in a lot of sense, they find, right, they're finders as well. But a lot of students have trouble with this idea of what are antibodies and what are antigens. So I like the way the book has called them finders and identifiers, because that's a big part of the problem. You have a lot of cells, different cells in your body, yours, number in the trillions, and then all the different pathogens, and then your own microbiome. Trillions and trillions, 100, 500 trillion cells. Just a lot. And then you consume cellular material. How does your body figure this out? So again, this is a big deal, antigens, and the use of antigen receptors to figure out friend or foe. All right. I say also types of lymphocytes, and again, plasma cells are part of the specific, okay? Natural killer cells are not. So plasma cells were what B cells became, so that's why you see that arrow there. And this looks different. It is somewhat different from a B lymphocyte, that's why we call it something else. But this is the actual cell that makes antibodies, and antibodies are part of the specific. Antibodies are proteins, but they are proteins that have antigen receptors. Natural killer cells, which is a great name, right, are these larger looking lymphocytes, and they do have visible granules. They're not as common, and we didn't point them out on your slides. Okay, but they're still, again, not part of the specific because they do not actually have antigen receptors. All right. Now, the primary lymphoid organs in lymphocyte development, well, when we call you a primary lymphoid organ, well, you're an organ that actually makes the cells. So that falls to the red bone marrow and the thymus. So red bone marrow produces, because that's where the hematocytoblasts are, produces B cells and T cells, but the B cells will stay, and they will undergo most of their development, which again has to do with gene expression, and they become able to defend you here. The T cells do not. They're born here and they move to the thymus, and here's where their gene expression is adapted to your body and your and the situation of your life. And I said again, they're controlled by hormones such as thymosins that we mentioned back in the chapter on endocrine. There are just trillions of lymphocytes in the body, and this has to do with the fact that if you have all these different cells I just mentioned on the last slide, trillions upon trillions upon trillions that belong in you, that don't belong in you, do they belong in you? So this question of friend or foe, right, comes down to presence of antigens. Well, now you have to factor in that on each cell, friend or foe, there are hundreds of different antigens. And for a B or T cell to be specific, they have to have the antigen receptor, which means that there are millions of varieties of B and T cells. Again, they all look the same to you guys because, nice round nucleus and sliver of cytoplasm. The difference is in their molecular aspects. What is on their surface? Their antigen receptors. So lots of variety. If you're going to have a good immune defense, you need a lot of variety. So here we look at the thymus gland in lecture and in lab. Pretty typical histology. This is just a cartoon to show that. This reminds you that it sits in the mediastinum, just anterior to the heart. It's very large in infants, and so your fetal pigs, it was very big, but it degenerates and is replaced by other connective tissue through your life. It's divided structurally into lobules, which is what this connective tissue divides it. Progenitor cells, so this is the precursor or the before cells, immature cells, arrive from bone marrow and mature and divide here. So we see areas where they're actively dividing and areas where there's mature ones that will then leave the thymus to populate secondary lymph tissue. So secondary lymphoid organs, right? So again, develop and mature in the primary, leave from, and do your defense from a secondary. They're called naive because they, again, haven't been exposed to the antigens on the pathogens yet. So these lymphocytes circulate in the blood and lymph and come to live in lymph nodes, spleen, lymphatic nodules. So that's our secondary lymph organs. So what do these have in common, these secondaries? All of them have lymphoid follicles, sometimes called nodules, but follicles and nodules, same thing, which have specific B and T cell areas. They have a germinal center, which is a site for lots of mitosis. Right? That's what germinal means. They have a post-capillary vessels, which allow cells from the blood to enter tissue directly. So again, cells from the blood, which would be other cells to come in and help, or we'd also be big enough to allow pathogens in. So when we look at the lymph nodes, and we'll look at these in lab as well as lecture, lymph nodes are in line to actually filter lymph. So they filter whatever's come in from the capillary beds that's been exposed to the tissues. So that includes debris, cellular debris, or other debris, and pathogens. They consist of dendritic cells and macrophages, which assist. And so these guys can kill pathogens. They just don't know what the pathogen is. That's where the B and T cells come in, and these live here and can be activated, right, by that pathogen entering, and then they can help to specifically destroy that pathogen, not harm other cells in your body. So this is what's very important about the B and T cells, and why I constantly call them the snipers, why I call the dendritic macrophages and other non-specific cells the shotgun. Right, so specifically destroy. There is a difference in the number of efferent and afferent. So the afferent, bringing it towards, there's always more of these vessels than the vessels leaving, the efferent. And this helps to slow down flow. I say it's like going from, on 75, you had 4 lanes of traffic and suddenly it's down to 1. You see what happens to flow, right? You probably lived it. But the idea is, again, we want to slow down fluid moving through here, fluid containing pathogens, so that there can be an encounter between these specific recognizers, B and T cells. All right? The lymph nodes are scattered throughout the body very strategically when you think about it, though. Not as many in the arms and legs, because the arms and legs, right, do not house vital organs and structures. So my defense does not need to be as strong here. It needs to be very loaded in this area here, where there's plenty of vital organs. They are generally in groups and chains, as you see on the models in lab. We have a number of regions that you can identify on those models, and again, really be thinking abdominal pelvic cavity and thoracic cavity. Right? The spleen here resembles a large lymph node, but is not in line with the lymph. It doesn't filter lymph, it filters blood. Right? So it has these large spaces in it that have blood in them, because again, the spleen is also, it's removing worn out blood cells, so it's perfectly situated, receiving all that blood to be able to determine whether or not we have a problem. All right. This time, though, the tissue itself is divided into red and white pulp. The white pulp is the nodules or follicles we talked about with lymphocytes, while the red has fewer lymphocytes, more blood cells, and that's why you get this contrast in color. And this, of course, is the largest lymphatic organ found in that upper left quadrant. We'll end this video here and pick up on the next video the rest of the structures.